Hello and welcome PGDMLT students. In this explainer, we're going to take a journey through the fundamentals of your applied histopathology and cytology course. We'll follow a patient sample from the clinic all the way to the final slide that can literally change their life. Let's get started.
So let's start with a really fundamental question, one that's at the heart of pretty much all modern diagnosis. How do doctors actually see a disease? I mean, really get a look at it on a cellular level?
Well, for us, the answer is histopathology. It's this amazing field you're about to dive into, and it's this perfect blend of precise science and, a bit of art. It's where technique and interpretation come together to reveal secrets hidden inside the body.
And make no mistake, this isn't just another lab procedure. For so many conditions, especially things like cancer, histopathology is what we call the gold standard. It's often the definitive final answer that tells a doctor exactly what they're dealing with.
So here's our roadmap for today. We're going to follow a patient sample through 5 major stages. We'll start with how we get it, and we'll end up with that final, beautiful slide that tells the complete story.
Okay, let's dive right in. First things first, it all begins with getting that crucial piece of material to work with.
Everything starts with a biopsy, and that's really just the clinical term for taking a small tissue sample from a patient for us to analyze. An MRI or a CT scan can show a doctor where a problem might be, but the biopsy is often the only way to know for sure what it is.
Now, right away, it's super important to understand the 2 main branches you'll be studying. We have histopathology, which looks at the big picture, how cells are organized into tissues. Then you have cytopathology, which zooms way in to look at individual cells or small groups of them. Both are critical for a full picture.
And of course, there are lots of different ways to get these samples. We might take just a small piece or remove a whole lesion. We can use a fine needle to pull out cells or fluid. We can even just scrape cells from a surface, like with a pap smear. And now there's even something called a liquid biopsy, which can find clues right in a patient's blood.
Okay, now for the part that feels a little bit like magic. We've got this soft, fragile piece of tissue. How in the world do we turn it into something solid that we can actually slice? This is where the core work of the histology lab really kicks in.
It all happens in 3 main stages. First, we have to get all the water out of the tissue, and we do that with alcohol. Next, we use a solvent to get the alcohol out. And finally, we soak the whole thing in hot, molten paraffin wax. When it cools, we have a solid, stable block that's ready for the next step.
And this picture? This says it all. This is exactly why every single step in that process has to be perfect. One tiny mistake, like cooling the block down the wrong way, and you get these cracks. We call those artifacts, and they can make the tissue impossible to read, which means a delay in diagnosis for the patient. Precision is everything.
All right, we've got our solid wax block. Now it's time for one of the most technically demanding and, honestly, one of the coolest parts of the entire process. It's time to slice.
We don't just use a scalpel for this, oh no. We use a highly specialized instrument called a microtome. The name comes from Greek and it literally just means small cut. And believe me, it does its job with unbelievable precision.
The goal is to create something that looks just like this, a perfect, unbroken ribbon of sections. And get this, each individual slice is only about 3 to 6 micrometers thick. That is significantly thinner than a single strand of human hair. It's a real display of skill.
And this slide right here shows you the difference between getting it right and getting it wrong. On the left, you see that beautiful, clean section. Everything is clear. On the right, it's a mess of folds and tears. Those artifacts can actually hide the very cells the pathologist needs to see, and that can have serious consequences.
So we have our perfect, ultra-thin slice of tissue floating in a water bath, and we've mounted it on a glass slide. But there's one big problem. It's basically transparent. To see anything, we have to make the invisible visible. We need to add some color.
So you might be wondering, why do we have to stain it at all? Why can't we just look at the tissue the way it is?
Well, the answer is pretty simple. Contrast. Without any stain, that tissue under a microscope would just look like a uniform, dull gray haze. Staining is what makes all the different cellular structures pop.
The most common stain, the absolute workhorse of every histology lab, is the H&E stain. It's a fantastic pair of dyes. First, you've got hematoxylin, which is a basic dye that loves acidic things, so it stains the cell nucleus a deep blue or purple. Then, you've got eosin, an acidic dye that stains the rest of the cell, the cytoplasm, a bright pink or red.
And this is what you get. It's beautiful, right?
A vibrant, high-contrast image where all the different parts of the cells are easy to tell apart. You can clearly see the blue nuclei against the pink cytoplasm. This simple technique unlocks a world of diagnostic information.
Okay, finally, let's talk about the principles that hold this entire process together: quality and safety. And after that, we'll take a quick peek at where this incredible field is heading. Look, this is probably the most important thing to remember. Quality control isn't just one step at the end. It is a system of checks and balances at every single stage. From making sure the patient's name is right on the sample jar, all the way to the final slide, preventing errors is our #1 job. And this field is always evolving. It's so exciting. We're developing powerful new techniques like immunohistochemistry, or IHC, which uses antibodies to light up specific proteins. We have molecular techniques that can find specific bits of DNA, and the whole field is moving towards digital pathology, scanning slides so they can be viewed and analyzed on a computer screen anywhere in the world.
So, as you begin your journey in this course, I want you to never forget the profound impact of the work you're going to do. Every single slide you prepare tells a patient's story. You are the skilled technicians who help uncover that story, providing the clear answers that doctors need and that patients are waiting for. Good luck.